morning, students. I hope you all are doing well, and today we will study the second part. The second part, continuation of the previous one. Application of biotechnology in health and agriculture, part 2. Subject teacher Imenungba. If you have any doubt, you can directly contact me. I have tried my level best. If there is any shortcomings, please do excuse me. Since we have studied the previous one, application in plants, that is in agriculture, today we let us study on biotechnological application in medicines. The medicine that we use is derived by the use of biotechnology only. And biotechnology is based on recombinant DNA technology processes. The recombinant, that is rDNA technological processes, has made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. That means for production of various kinds of medicines or drugs, we use this technology, biotechnology, that is recombinant DNA technology. Let us learn about insulin today. Genetically engineered insulin. This is very common thing. Have you ever heard about diabetic patient? Yes, this diabetic patient are asked to have insulin injection. When this pancreas fail to secrete insulin, one starts suffering from diabetes or sugar, and so this insulin has to be injected into the diabetic patient. When we have a look on an adult person, it can be controlled by having insulin at regular intervals. The main source of this insulin was isolation of insulin from animals. That means from the animals only it was isolated before. Nowadays, insulin can be obtained from bacterium using technologies of biotech or biotechnology. By using this biotechnology process, we can obtain it from the bacterium or bacteria. This is more safe. Insulin was early extracted from pancreas of slaughter cattle and pig, but insulin from this source developed allergies or other type of reaction to the foreign proteins. When we have a look on this, in the olden days, insulin were extracted directly from the slaughtered cattle and pig from the pancreas. But they turn out to be cause various kinds of reactions, or it may cause allergy. When we have a look on insulin consists of 51 amino acids. Keep in mind, 51. That means and these 51 amino acids are arranged in two short polypeptide chain, that is A and B, that is pairing 21 and 30 amino acid respectively. That means chain A will carry 21 amino acid and chain B will carry 30 amino acid, and these two, chain A and B, are linked together or interconnected by disulphide bridge. This is the structure of insulin, which I have explained just now. When we have a look on in human being, insulin is produced as a prohormones. Prohormones which contain an extra stretch of C peptide. When we have a look on the previous diagram, you can have a look. They contain an extra stretch of C protein, which has been insulin, which is being produced in human, which is absent in measured insulin. The main challenge for production of insulin using recombinant DNA technology techniques was getting insulin assembled into a measured form. An American company known as Eli Lilly in 1983 prepared two DNA sequences corresponding to chain A and B of human insulin, and then what happens, and then introduced into the plasmid. That is Plasmid means something like nucleus, okay, into the E.coli, that means asterogen E.coli, that is a bacterium, that is been after they've been produced, they have introduced this one into the bacteria to produce insulin chain. Okay, chain A and B were produced separately. After they have been produced separately, it has been extracted and combined by creating disulphide bond to form human insulin. And this human insulin, we call it as humulin. Okay, humulin. Okay, next one we have got is gene therapy. Okay, gene therapy. As we all know, gene therapy means manipulation of genes. Suppose if I have got defective genes in me, that gene can be taken out and replaced with a perfect one, nice one. Okay, and this has been using for treatment of various kinds of disease. Okay, gene therapy involves treatment of hereditary disorder caused by single defective genes by replacing the defective genes by normal genes. In this method, genes are inserted into a person's cells and tissues to treat a disease. Okay, we have got two types of gene therapy. Germ-line therapy and the other one is somatic gene therapy. Okay, when we have a look on the first one, germ, that means in this type of therapy, functional gene is introduced into the germ cells, that means into our reproductive organs or reproductive cells, that is sperm, ovum or zygote. Okay, somatic cell means apart from the reproductive cell, other part. Okay, we call that one as somatic gene therapy. The functional gene is introduced into somatic cells, and we are going to study about this common one, that is ADA deficiency now, or else we can call it Severe Combined Immune Deficiency, SCID. Okay, let us first look on the somatic gene therapy. In this, okay, the first trial, okay, we call it as adenosine deaminase deficiency. Okay, this was first done, okay, this was gene therapy was first done in 1990 to a 4-year-old girl who was suffering from adenosine deaminase deficiency. This disorder is caused due to the lack or due to the deficiency or due to the deletion of enzyme adenosine deaminase, and this adenosine deaminase is very much required for our immune system to function properly, and this defect can be treated, in which functional ADA is given to a patient by injection or through bone marrow transplantation. If this disease is not treated properly in time, it may prove fatal. The treatment of deficiency involve various means. Okay, we have first one is called transplantation of bone marrow with normal ADA genes. That means through transplantation of bone marrow. Next one is enzyme replacement therapy, and this is done by injecting functional adenosine deaminase enzyme. Third one is gene therapy by introducing functional ADA gene into the lymphocyte. Okay, that are the three ways. Okay, when we have a look on this gene therapy method, lymphocytes from the blood of the patient are grown in culture medium outside the body. Okay, and what happens is that a functional adenosine deaminase recombinant DNA is then introduced into the lymphocytes and returned to the patient. In this method, periodic infusion of such genetically engineered lymphocyte is needed. If gene isolated from the bone marrow cells producing ADA is introduced in cells at early embryonic stage, it could be permanent cure. Okay, this is the one that is all for today. Thank you.